everyone. I'm Smita Krishnaswamy. I'm from Yale. Today I'll be talking to you about how to understand very high-dimensional, high-throughput data. I mostly work on single-cell data, but also other data types that this could be applicable to. And I'll mainly be talking about sort of the machine learning mathematical techniques that we use to really understand and interpret this kind of data. And broadly speaking, we call it manifold learning, but I'll go more into specific techniques we use for that as I go through the talk. So the main challenge that we're dealing with is very generic. It's basically that we have very high dimensional data. High dimensions refers to the fact that we have a lot of features in our data. So in the back of your mind, you want to think of something like single-cell RNA sequencing data. So if you have single-cell RNA sequencing data, the features are the gene expression counts for each of your gene. And so how many dimensions are there? Let's see who's awake. How many dimensions? Yeah. Who said that? 20,000. Yeah, about 20,000 or more. That's how many dimensions there are. And it's also high throughput. High throughput means there's just a lot of observations, because in single-cell experiments, you get on at least thousands of observations, possibly many more observations these days. There's some data sets that 10x and Human Cell Atlas have put up that have around 2 million observations. And of course, because these are cells, they're living entities, they're changing, growing, they're all different from each other, it's very heterogeneous. And so just taking this kind of data and making conclusions about what's going on, inferring mechanistics like how the gene regulatory network is working in a cell, how will a cell respond to stimuli, or whatever other biological insights, sometimes descriptive, that you want to come up with, tends to be a challenge. And the main challenge you can think of as how do we really look at this data? You know, first off, humans can't really look in more than 3 dimensions, and we have 20,000, so how do we really understand what's going on in this data? It's far beyond our intuition, but that's not it. This data has a lot of sparsity, so there's a lot of incorrect or missing counts. It has a lot of noise, potentially artifact, so if you have single-cell data measured in different batches, you might have batch effects that make them completely unaligned. A lot of the effects are nonlinear. So in some sense, if a cell is responding to something, it has to be nonlinear. It's not going to go up forever, right? It has to be capped. So there's a lot of nonlinearity involved. And just the scale of the data can give you computational problems, which I heard some people addressing earlier today. So we have a lot of challenges, and based on this slide, you might think, oh, this is pretty hopeless. We should just give up. But actually, you don't have to give up. It turns out that, like many things in the world, even though it has very high-dimensional ambient space or high-dimensional measurement space, a lot of cellular systems actually lie in a much lower dimensional subspace. So the classic example of this that actually every manifold learning sort of paper or slide has to have is the Swiss roll. But I was told by a very prominent Swiss scientist last week that in Switzerland there's no such thing as Swiss rolls. So I don't know where they came from either. So anyway, the Swiss roll, you see that it's embedded in 3 dimensions. But depending on how you think about the noise, it's either 1 or 2 dimensional when you unroll it. So that's a smaller dimensional object kind of rotated and living in a larger dimensional world. And that's the kind of picture we have of how to understand cellular systems. And you might be wondering why cells would have this kind of structure. It's because the genes that are in a cell have to be coordinating. They have to be making proteins that interact with each other that eventually lend themselves to making a very coherent entity, which is a cell. And so there has to be informational redundancy between the genes, and so it cannot have intrinsically that high of dimension, aside from some of the noise. So just to give you a more visual example, if there was no structure, it might be spread through all the dimensions. Now imagine 3 is 20,000, like this. If it's like this, there's not much pattern there to analyze or assess. Because there is this redundancy, informationally speaking, between the genes, and because the cell types are constrained to sort of viable cell types of living cells that can function, you have a much smaller dimensional space and a much higher dimensional object, so they're not spread evenly through all the dimensions. And a lot of what I'm going to talk about today is exploring this kind of data, coming up with hypotheses and insights about this data that's fairly unbiased by just learning this shape in a non-parametric way. And I'll show you two main techniques for learning it. Not that you're going to perhaps not become an expert on these techniques, but you'll know that there's sort of two classes of techniques that we could potentially use. And we use them almost equally in my lab, and these include graph signal processing techniques and deep learning. And both of these can be used to, in sort of an unsupervised way, learn this intrinsic shape of the data that I'm showing you here. And then using these, I'll go over several applications, but conceptually, all of these applications are in some way related to what I said about manifold learning the intrinsic shape. So if you do manifold learning, you can do things like denoising, visualization, comparing between experimental conditions, and comparing large batches of data with a neural network in a scalable way. So I'm going to get started on the first type of technique, which are graph signal processing techniques. And here, in at least one of the examples, I'm going to use a cellular system. And just to give you a little bit more insight on what the manifold structure might be here, so we're going to use as a running example in the first part of the talk, breast cancer cells that are undergoing an epithelial to mesenchymal transition. So it means they're going from epithelial to mesenchymal states, but the cells sort of lie somewhere in this space of epithelial, intermediate, mesenchymal. So the transitions between the cells are in small steps. They're not sort of drastic. And you can actually find cells everywhere in between, even though sometimes in the past biologists haven't studied everywhere in between. And so this kind of smoothness actually allows you to learn this shape. So if you had big gaps in this data structure, you couldn't really learn the shape, but because it's fairly smooth, you can learn it. So the first way I talked about was graph signal processing. For this, basically, you're modeling the data as a graph. And a graph can follow the manifold structure, and it can follow it really well depending on how you make the graph. The main idea here, and I know Donna talked about graphs, but we'll amp it up with some signals on the graph next. So if you have data, for example, in 2 dimensions, the way you make a graph out of it is you make a weighted graph that's weighted by the distance between these 2 points. And then you pass it through a kernel. So things with high distance have low affinity. Things with low distance have high affinity. And when you pass it through that kind of kernel, it zeroes out the edges that cut across the manifold. And you're really almost left with something that looks very much like the intrinsic shape of the data or the manifold. But what enhances this is if you Markov normalize this affinity matrix and diffuse through the graph. So if you started walking along the graph, seeing where these paths would lead, you don't actually have to walk, you can just power a stochastic matrix to do this, then you'll connect long-range distances in the graph. So now once you make this kind of a graph, there's a lot of different things that you can do on graphs that are very successful. There have been labs that have done a lot of clustering. We've done visualization, denoising, comparing between experiments. But just to give you a little bit more insight on what's happening, the process of diffusion that I described to you was first invented about 12 years ago or 14 years ago by Rafi Koifman, who also happens to be one of my colleagues at Yale. So the main idea here is if you create a graph in the way that I showed in the previous slide, by diffusing through the affinity-based graph and creating longer range connections, then you have a matrix that's called a diffusion operator. Now if you eigendecompose, how many of you are familiar with eigenvectors and eigendecomposition? Okay, good. A lot of people are. So then the eigendimensions of this diffusion operator actually give you the intrinsic dimensions of the graph. And this eigendecomposition is called a diffusion map. And the diffusion map has, so it's basically taking the diffusion operator, eigendecomposing it, and everything but the main non-trivial eigenvector gives you a dimension that's important, that's intrinsic to your data, that's nonlinear. So it can find the curve in the Swiss roll as one of the dimensions. So this is a form of nonlinear dimensionality reduction, and it turns out nonlinear dimensionality reduction is very important for manifold learning. So what else is really cool is that the eigenvectors actually form frequency harmonics in your data. So how many of you have heard of Fourier transforms? Okay, so Fourier transforms are a way of decomposing a signal into cosines and sines. It turns out you can decompose a signal in a graph also into cosines and sines. You don't have to use cosines and sines. You just have to use the eigenvectors of the diffusion operator that I just showed you, which would be similar to the eigenvectors of a graph Laplacian for people who are interested in graph spectral theory. It just turns out that the eigenvalues are switched in order. So you see that the first or second eigenvector of the diffusion map corresponds to low frequency trends. So you see the value of the eigenvector. I don't understand what that black thing is. The value of the eigenvector slowly goes down. And the second to last one is describing a very high frequency trend. So these eigenvectors are giving you frequency harmonics over the graph. And so you can think that you need this because usually Fourier transforms happen over time or space, which is some kind of continuous setting. Now you're taking that to a more discrete setting where your space isn't continuous, so you have a graph as your base, and you're really looking at sines and cosines over the graph, which are eigenvectors of this. So we will use these concepts that these frequencies are represented by the eigenvectors in order to do several things. So just to, again, bring this back. So you have a graph that you created, perhaps using the diffusion-based method that I said. You have signals on a graph. For example, a gene value can be thought of as a signal on a graph. So each node in this graph is a cell. These cells are connected to one another based on the affinity matrix creation. And then now you can take the gene value in a cell and think of it as a signal on the graph. It could be high or low. In this particular example, they're showing negatively valued signals, which we may not have. But these are signals on the graph, and this is how it works. And much like you can take a Fourier transform, you can take a graph Fourier transform, which basically means load the signal into the eigenvectors of the graph. Once you load the signal into eigenvectors of the graph, you go from a vertex domain representation to a Fourier representation. So this is a graph Fourier representation. The low frequency components of this representation represent a low frequency varying trend, and the high frequency are noise, like in Fourier harmonics. So just in case you're kind of familiar, here's the difference. So in a regular graph, you could have signals or some kind of time-varying observations, but here signals are features of your data points. Signals in regular Fourier analysis, regular signal processing might be some signal over time. Here we have signals over a discrete point set. There you would decompose into sines and cosines, but here you're decomposing into eigenvector bases of graph Laplacian or equivalently the diffusion operator. Filtering a signal on a graph is modification of the Fourier domain. So if you have a low-pass filter, which usually gets rid of noise, then you can do the same thing. You can get rid of noise using low-pass filters on a graph harmonics. There's some things that don't translate exactly super well, but people have been working on it. There's a whole conference on graph signal processing if you're interested in this. The graphs tend to have a less clean notion of translating signals, but you can still do it by convolution with a Dirac. And then, so you can convert back and forth using the eigenvector space and the conjugate of it to go back and forth. And so with that, I'm going to talk about one of the algorithms that we developed. Actually, when we wrote the paper, we didn't really write about it in a graph signal processing way, even though we mentioned the connections. But since then, I've become enlightened to graph signal processing, so I'm going to explain it from a graph signal processing perspective now. This was joint work with my postdoc soon to be faculty member David Van Dyke and also some people from Donna Pierre's lab. And the main idea here is that we can use this idea of signal processing to clean up the signal and recover from something that's called dropout. And in the Vertex domain, it translates to cells learning from each other. So one of the first things that happened when I used to work on CyTOF data back in the day, which was 32 dimensions and many thousands of cells, but when single-cell RNA sequencing came out, it was really exciting. I was like, oh, there's so many more dimensions, and now we can learn the gene regulatory network. But it turns out you couldn't really, and the reason is because most of the values in a gene are dropped out. And so you have very little information left at the gene level. So there are some people who talk about should you correct gene values or should you leave them alone? And the question is, do you want to learn gene-gene relationships or not? So some people pose it as kind of a philosophical question, which is a practical question. If you don't want to, if you just want to learn clusters or something, then maybe it's okay. So again, just to belabor the point, I'm creating the same graph that I mentioned. I have the original data. I calculate pairwise Euclidean distances between cells. I pass them through an affinity matrix. We Markov normalize and power this affinity matrix to get rid of spurious connections and clean this up. So, and then the next thing that we do is we actually multiply the gene expression matrix by this, which actually turns out to just be loading. When you power the affinity matrix, you're getting rid of the high frequency eigenvectors, and you're basically loading the signal onto the low frequency eigenvectors. So that means you're low pass filtering the signal. And that's basically what we're doing in MAGIC. And I'll explain it from a filtering perspective in a second. That's how it's explained in the paper. So before MAGIC, you have something called dropout, because about 90% or 90 to 95% of the transcripts are missing in single-cell RNA sequencing data. Some people think entries in the matrix are missing. Entries in the matrix are not missing. It's actually counts that are missing. So this is some kind of regular noise that's dropped out from the data. So actually, I would call MAGIC more of a denoising algorithm that just gets rid of this dropout noise. And after MAGIC, you see how you have much clearer structure than you even had before. So the main thing that's happening is when we diffuse the matrix, what we're doing is we're taking this signal on the affinity graph and we're low-pass filtering it. So as you increase the T, you're getting rid of the high Fourier components of the graph. And you're retaining the low frequency components, which gives you a smooth signal. And the idea is similar cells should have similar transcripts. And so if there's some kind of high frequency change, that's noise. And we directly get rid of it the same way you do in EE, which is the low pass filter. And when you do this, you have something that looks like this, where it's hard to analyze if there's a relationship between these two genes, whereas people who know EMT know that there's a strong relationship between those two genes. And as you clean the signal more and more, you start to recover the potentially nonlinear dimensions, these nonlinear relationships between the dimensions. And this is our version of imputation, which is not filling in missing values, but denoising the data. And how do you know this is right? So whenever you have this kind of computational technique, people think, oh, we even called it MAGIC, so maybe we're making it up. So hopefully we're not making it up. And we usually tend to validate things both computationally with a test case where you know the ground truth. Sometimes in biology, I feel like the reviewers don't even understand that you need to do this. You need to have a test case where you know what it looks like, and then you need to show that your technique works on that, and then experimentally with an experimental follow-up. So computationally, we took this data set that doesn't have much dropout because it's actually bulk RNA-seq of these very small worm embryos. They have natural manifold structure. We randomly dropped out 80% of the signal. And then we imputed it back with MAGIC, and we can see the relationships are restored. Sometimes they're even cleaned up. And we report statistics on this in the paper. And then the next thing that we try to do is, the reason we wanted to do this so badly was we wanted to learn gene-gene relationships. So then we see, can we predict correct gene-gene relationships after magic? So the main idea here is that there are so many genes in this data, which are columns, and do these columns have mutual information between them, which would imply some sort of co-expression relationship, or target, source-target relationship, like between a transcription factor and its target. And so we sought to predict the latter. So we use an information theoretic metric that I developed back as a postdoc, which is a density resampled estimate of mutual information, because this data tends to be very highly bias sampled. So we basically renormalize the data and then compute the mutual information. And when we did that, we looked at a transcription factor called ZEB, which is known to be involved in EMT. We can tell that there are some genes that increase after it. So there's a source-target type relationship. And then after after magic, we compute the DREAMY metric. And then we show that these genes are actually immediately turned on by ZEB by doing two different kinds of inductions. You can also, you can induce EMT directly by turning on ZEB, or you can do what we were doing, which was feed it with TGF-beta. But if you directly induced it by turning on ZEB, you'd skip a whole bunch of those, and you'd get the genes immediately that we predicted. And we can show that to a high degree of significance, that the rank genes based on the expression of TGF-beta versus ZEB, and that the difference in rank in the two expressions are pretty high. And so this gives us some confidence that we actually predicted the right genes here. But of course, there's other ways you could sort of validate this, like if you had a knockdown or something, but we just didn't. So the next thing that we wanted to do was, magic was cleaning up one data set. But as single-cell RNA sequencing became more and more common, we wanted to compare two or more data sets, because people now are comparing, experimental and control, healthy disease, or multiple time points. And in these cases, this is also a problem in single-cell RNA sequencing data that people maybe haven't carefully considered. It's a problem of organizing, sort of larger the experimental setup more than what you have. So you want to tell the difference between the conditions, but you have to go through the single cell data to do it. And there can be many challenges here. There's weak effect sizes, there's batch effects, there's a problem integrating the data. So we sought to work on these. So first we worked on a method called MELD that actually Dan over here largely worked on, along with some other students and postdocs in the lab, for carefully comparing two or three different data sets. So in these examples, I'll go over how to compare two data sets in single cell and what the problem is, like why is there a problem. And we'll use, of course, graph signal processing, because I set it up that way. And it wasn't just posted on bioRxiv, it's been there for a while, but that slide's old, sorry. You can look at it on bioRxiv for sure. And so the basic question is, if you take just some example, you take cells that are stimulated and cells that are unstimulated. How can you tell the effect of this stimulation? If you measure it at a bulk level, you might see some effect, but at the single cell level, many of the cells might be affected, some of the cells might not be affected, and you want this information, otherwise you wouldn't have measured it in single cell, you could have measured it in bulk. And how can you tell when it's just noise that's different between the two experiments versus the signal that's actually different? Why is this advancing? And so when you look at the overall bulk level expression differences, they can be pretty weak between the two conditions. But it turns out that there are sometimes very important differences between cell types. So, but the problem is that the effect size is small relative to technical noise. Here you have the stimulation and unstimulation condition in green and blue, and you see there's a large degree of overlap between the cell types that occur. It's not like the whole population shifted over. So you often have this where it's weak. I think you did this too. It's like automatically advancing, so I have to keep going back and forth, sorry. And so what we're trying to come up with is sort of a holistic view of this. So we want to take this combined data and we want to say, throughout the whole manifold, what areas are enriched in the experimental condition, what areas are enriched in the control condition. And this isn't just like, differential expression. I'm not bulking it up and just taking differential expression, but I'm trying to make a statement about the whole cellular manifold, which you can learn with single-cell RNA sequencing data. So the main idea is the control cells might be distributed high-dimensionally in some way through the manifold, but after perturbation, you have a change. The change can be subtle. It can be in certain areas. And we want to take, the whole manifold into account when we tell you what's different between the two single-cell populations. And this would involve saying they're different exactly here at whatever level of granularity that you want. So the effect of the experiment is essentially another sort of differential signal that tells you at each point in the manifold, is this more likely to occur in the experimental condition or in the control condition. So you have this third kind of variable or signal that tells you for each cell type or cell point in the manifold how different they are. So here we have the experimental. So in order to do this, we go back to our graph signal processing where we have the experimental label as a signal. So how do we do this? We perhaps use batch normalization. We just combine the two experimental conditions into one graph. The same way that I told you before. And then if the cell is originally from the control, we give it a +1. If it's from the experiment, we give it a -1. So we created what's called a raw experimental signal. And now we low-pass filter this signal by paying attention to its frequencies in different places in order to get a smooth signal, which basically gives you an estimate of the likelihood that it's from either control or experimental. So so the basic idea here is you can learn the graph from the single-cell profile using the Gaussian kernel-based graph construction. You add the raw experimental signal, and then you filter it. And our filter is worded as a constrained optimization here. Instead of a fixed low-pass filter, we're trying to recreate the signal if it naturally varies a little bit as much as possible while eliminating noise and taking sort of point density estimates of it. So what that does is when you take such a filter, and you can solve that optimization in several ways, you can have this true effect. You can have an added contamination, and you'll get a composite effect. But when you filter out sort of the contamination, you should get the true effect back. And that is Minnesota. I see some people are from Minnesota. The graph signal processing world is filled with Minnesota road maps. So you're awesome. So I don't know why. Do you know? Just because Schumann is there. So after you have this signal, what can you do? You have this filtered signal. What can you do? The one thing you can do is interpolates the signal, just going from a discrete signal to more of a likelihood that goes from 0 to 1. So now you can find genes that track with the experimental condition, perhaps what's driving the experimental difference, by regressing against this new signal. But what we can also do is what I originally said. We can pick out areas in the manifold that are changing with respect to the experiment, even if they're small, even if they're disconnected parts of the manifold. And we do this by another algorithm we call vertex frequency clustering. So basically, we take the Fourier transform of the low experimental signal, the graph Fourier transform, and we look at its frequency characteristics, and we realize that if we clustered by this, we would get what we wanted. And this is more work that's done by Dan and Jay. So here we see that all the cells at the top are from the control condition. All the cells on the bottom are for the experimental condition. Then you have a slow transition between them. So these cells are transitioning this way. But then you see the cluster on top. Do you think there's a slow transition in that circular cluster on top? So no, there's not. It looks like that cluster just doesn't respond to the experiment because it's evenly mixed. So what we notice is that if you have a high frequency raw experimental signal, then that region isn't really responding to the experiment. Those cells stay the same and they have an even mix. So they quickly change from 0 to 1. And in areas where it's really changing, there's a gradual change like this. And we can pick out exactly these areas that are enhanced for the experimental condition, areas that are slowly changing, areas that are enhanced for the control, and areas that are not responding using vertex frequency clustering. And then we can give you exactly what I said, all the areas of the manifold that are changing in response to the stimulus. And again, the way we do this is that we add this MELD filter, which gives us this enhanced experimental signal, and we use it to pick out areas on the manifold. And of course, the plots that I'm showing are PHATE plots. And how much time do I have?
14 minutes.
Okay, 14 minutes. I'm speaking fast, so I'm getting through stuff. And then the other point that I wanted to make is that this signal has value on its own in terms of picking out things that might be driving the differentiation. So when you do this on this T-cell differentiation network, you indeed see that things that have to do with T-cell response, activation, and things like that, that you couldn't necessarily pick out before because you have this more discrete signal and discrete distribution that you aren't sure. You can do some t-tests or something to see if they're different, but here you clearly see what signal tracks with what, because as you increase the experimental signal, you have a sharp trend up or down, which again you can quantify by mutual information. I'll sort of skip this. The main idea here was that if you do use vertex frequency clustering on this signal, you can basically pick out small areas. Whereas in this, so maybe I'll go over it briefly. In the main experiment, they had done clustering first, which a lot of people tend to do. They cluster single cell data set 1, single cell data set 2, and they compare cluster proportions. But that doesn't really say anything because the level at which the two experiments are different could have nothing to do with the granularity of the clustering. And in fact, that's what we see. This was published work, this from the Alan Klein lab. That uses zebrafish development, and they had some kind of clustering that they'd done, but you see if you look at it, the clusters have different sort of proportions. So there'll be some part of the cluster that's enriched for experiment, some part that's enriched for control. And when you look at it, the cluster level is not good to say. So if you say that the last to one cluster has more cells from the control condition, it's a little bit misleading because only a part of that cluster has more cells from the experimental condition. So we show that our more fine-grained vertex frequency clustering can rectify that. And if you take that very cluster that I showed you, you can see that vertex frequency-wise, it has a lot of subclusters, and actually only one of the subclusters, the somite subclusters, is really more enhanced in the knockout condition than in the regular condition. So this is the kind of thing that if you don't want to cluster first and then compare, you really want to holistically compare the manifolds and then come up with the areas that are changing, which is what we do using our graph signal processing method. The sort of final two topics that I wanted to talk to you guys about is what is the visualization that I've been showing over and over again. I probably should have presented it first, but I'll present it now. But magic to meld is kind of a natural transition. It's PHATE, and it also uses the idea of graphs a lot, though perhaps not graph signals very much. The idea of PHATE is that we take global distances on a graph, so from one cell to every other cell, via diffusion affinity operator as the context of a cell, much like a word to vector type thing where you get the context of a word in a vector. And we use an information theoretic distance between these contexts and squeeze them into low dimensions, which gives us PHATE, which preserves local and global information. And we show that on these two examples. One is an artificial tree, and one is an embryoid body data set. The artificial tree, we just drew it. And it's drawn like this. We rotate it into high dimensions and add noise. And then we see, can we visualize it again? That's sort of the computational validation way that we're doing it, although I'll explain it a little bit more. So when we take this artificial tree, and if you want to visualize it with PCA, t-SNE, and PHATE, you see that PCA can pick out sort of global distances because it gives the global directions of most variation. So actually it's okay at picking out global structure, but the local details like the branching it misses because it can't denoise in nonlinear directions. t-SNE feels free to shatter data because t-SNE's optimization really heavily favors keeping local distances, and it doesn't have actually much of a penalty. With the asymmetry of the KL divergence penalty, doesn't penalize much for putting far things close, but it only penalizes a lot for putting near things far. You can work out how the ratio in the KL divergence works. So that means it feels free to shatter things. And so PHATE, because we keep the global distance of every cell intact until the last step of PHATE, it keeps both global and local distances together. And for example, in our embryoid body data set, we had these embryoid bodies, human embryonic stem cells grown in embryoid body cultures for 27 days, taking samples every 3 days, and we measured them in 5 different single-cell RNA sequencing experiments. You see that the red cells that are in the beginning, they show up as a thin line because they're all human embryonic stem cells. They're one cell type, and then they're differentiating, so you see the divergent pattern. Okay, so you see this global pattern on PCA. Again, PCA is okay at showing global structure. TSNE does not show this global structure at all because it's randomly shattering the global picture. So if you don't want a global picture of your data, then you can look at TSNE basically, because it shatters data like this. And then if you, and UMAP does the same thing, more or less. And diffusion maps is the technique that I mentioned before that Rafi invented. And this technique can keep trajectories together, but it puts them in different dimensions, so it's not good for visualization. So one of the things that PHATE does, it corrects this tendency of diffusion maps to splay things out to different dimensions, squeezes it into 2 dimensions. And the way we do this is we take the data, we take distances in the data as we did before, and then the affinities. And then we diffuse them. But now we look at this whole probability structure and we say, what's the probability that one cell goes to every other cell? What's the probability that another cell goes to every other cell? So this is giving the global context of the cell. And these are 2 different probability distributions, and we take a divergence between them. Usually when you compute probability distances between probability distributions, you can't just use Euclidean distance, which it turns out that's what diffusion maps sort of do. So that's why they don't keep trajectories together. So we take this M divergence that's basically taking the pointwise log of each row and the distance between them, and then we embed it with MDS to keep the global structure. But it turns out that we don't have to take the log. We can take any information, theoretic sort of submodular distance transformation, which is square root or p log p. And then we've done a lot of comparisons. So we had to keep revising this because, you know, whatever, the reviewers don't trust me yet because I'm a junior PI or something. Now we have 1,200 comparisons in the paper, and I'm not joking, with all kinds of methods that they wanted us to compare against. And we usually think PHATE looks more like the ground truth, right? But, you know, looking more like something that you think isn't sort of definitive proof. So for example, here's PHATE on a retinal bipolar data set. As I showed you before, PCA keeps the global structure, so PHATE's keeping that same global structure, but it's showing more local relationships. And likewise, on our embryoid body data set, we can see a lot more structure, and we can find different intermediates in differentiation. We can draw this beautiful tree that I'm not going to go into. But we were thinking, how do we really tell that PHATE really keeps more global structure than other things? And this again has to do with ground truth. What we do is we create a really clean manifold trajectory type structure, or also cluster structures, various different structures that have no noise. When you have no noise, geodesic distance is equal to manifold distance. So then we add noise, and then we put it into high dimensions. And now we put all the methods through it to see if they recover denoised geodesic distances that we originally had. And we see that PHATE performs the best by far on these myriad of comparisons where we knew the ground truth. And so we encourage you to use PHATE as I've been. And then the final thing that I want to quickly broach, because I'm probably running out of time, is what to do to scale this kind of thing up, okay? So diffusion maps and graph construction scale to some extent. We actually just wrote a paper that we published in Sampling Theory and Applications that we can compress a diffusion operator by way of landmarks. But at some point, the scaling becomes an issue. And this is when you have many, many patients in a hospital measured in single cell. And we were actually encountered with a data set that had 186 patients that were all measured in single cell. And they had 20 million cells total. So you can't call these common algorithms like t-SNE or k-means or anything like that. So in order to get our things to, all these algorithms methods to scale, we started to use an autoencoder. So an autoencoder can nearly in the same way as graphs learn the manifold structure, and that's because of its informational bottleneck. So if you have an informational bottleneck, it's in the bottleneck, it's forced to make a meaningful representation of the data because the autoencoder is just penalized to reconstruct its input at the output. So if you set input equal to output, but you bottleneck its dimensions, it's going to learn something meaningful in between. And this is a powerful idea that Hinton and company and Salakudinov came up with in 2006. But the problem with most autoencoders and other neural networks is what they give you in between is not interpretable. It could be crazy looking, it could be anything. So in our lab, we have a lot of efforts and a lot of papers to make the internal layers of an autoencoder interpretable. And I'll go through one quick one. So this is Saucy. This is our paper. It's going to appear in Nature Methods. Saucy has a whole bunch of regularizations in the internal layer to take all of these patient samples, batch normalize all of them, which no other batch normalization tool does, and just read off cell types and then compare populations. So how do we do this? We do this via regularizations, and I'll show you just one of the regularizations, as I think I'm running out of time. So if you have a representation, so we wanted one of the layers to just give us what cluster is this in, cluster 5, cluster 6, something like that. So in that layer, we want a very binarized representation, very discrete representation that just says the number of the cluster the data is from. But it still has to recreate the signal, so it should be storing the rest of the information in small fluctuations around these large binary values. So we penalized it so that, for example, this cluster would be 1, 1, 1 with very strong activations, or this cluster would be 0, 1, 1, very, very weak activation and 2 strong ones. And so in order to do this, what we do is we treat the activations as probabilities, and we penalize the activation entropy. So it encourages activations that look like this rather than ones that are all the same level. So you see it gives you a binarized output. And this binarized output, you can just cut it and read off clusters. Another regularization we did, the one for batch normalization, is because the 2 patients that we put in due to batch effects, the embedding, the visualization you get from the middle layer, so it automatically does something like PHATE in the middle layer fits 2, are totally off from each other. So how do we realign it? So we give a probability distance penalty called MMD to realign them. So why does this realign rather than totally scramble, right? Why does it not scramble the data? Does anyone know the answer? It's because of the reconstruction penalty. So if it totally scrambled the data, then it can't reconstruct well because it still has to reconstruct the data. So it's encouraged to minimally realign rather than totally scramble the data. So after MMD, you see the data sets are aligned, and in fact, they're aligned by cell type. So then we can combine all these 200 patients. They were all dengue patients. And then we can do this type of an analysis where we see the acute combined manifold, the healthy manifold, and the convalescent for this infectious disease. And we came up with actually very interesting signatures of these dengue patients. Somebody told us that we were just using this as an example for our neural network that we liked so much and that we really should have written a different paper on this. But we're not experts on infectious disease, so hopefully somebody else will do that. And so we can come up with these signatures of the distance between these patients, and it turns out one interesting thing here is that gamma delta T cells, which people don't think of a lot, it seems to be a sign of an acute infection or a recent infection that sometimes is undiagnosed because these patients were all from India, so the medical care there varies. And the MMD distance between the whole samples can give us a way of embedding or doing something like PHATE on the whole population, and here each dot represents a patient rather than a cell type. And we show that because we use an autoencoder, Saucy's nearly the fastest one. The only one that's faster is PCA with randomized projections, which of course doesn't give you very, very good. And this is only in terms of, so in Saucy we had so many comparisons to do because we had to compare to clustering methods, visualization methods, and batch normalization methods. So if you take all of these methods and run their scalability, then randomized projected PCA for visualization is faster, but as I showed you, PCA misses all the details. So those are the 2 classes of things that we work on in my lab to learn manifold structure of data. We use graph-based ideas, and we use manifold, and we use neural network-based. Our neural networks are what are called these days self-supervised. So they're either cycle GANs that can supervise themselves, or they're these kinds of autoencoders. So I'd encourage you to sort of read widely also in that field in addition to single cell biology to understand more of these concepts. And if you want more links or something like that, feel free to email us. I'm going to skip over this, which is another cool regularization that we submitted to NeurIPS. But the main conclusions are that if we really learn the manifold or sort of the shape of the data, it enables us to extract structure and patterns from it. So when we use graphs and signals on graphs, we were able to denoise data and thereby impute values, enable low-dimensional visualization, infer experimental change dimension, And when we use deep learning, we're able to regularize the deep network so that we can examine the internal layers, which also can learn the manifold because of reconstruction penalties. And here are some of the few people in our lab that I'd like to acknowledge. So that's Kevin, Scott, and they're all at my house. And Dan's a really good cook, even though he's pretending like he's burning that grill. If you want any of our software, all of our software is on GitHub. We have a help Slack also if you want help using our tools. You can follow us on Twitter to see what other things we're cooking up. And we have an active website. I am looking for postdocs, so if you're interested in postdocing, let me know. And here's acknowledgement to all the people, especially the experimental people who gave me all this fantastic data. The Jim Noonan Lab for Neuroscience Data, Ivanova Lab, soon to be at University of Georgia, that gave us the EB data and help with it. The YCGA, which sequenced all of this. Math Health from Rafi Koifman, Guy Wolf, who's now in Montreal, who used to work with us, the Donna Pierre Lab, and all our funding sources. And if you have any questions, I can take them.